So here we're going to be talking a little bit about the mechanisms of innate immune responses. We already have discussed the basic functions of the white blood cells. Now we're going to talk like what you need to know. So innate immunity. We're going to structure the responses into several major categories. First is going to be inflammation. That's the mechanism of white blood cell response to the injury that we have discussed previously. So these white blood cells can respond in a way that they phagocytose pathogens and dead cells. Two main phagocytic cells are macrophages, that is designation for the macrophages, and neutrophils. The difference is that macrophages, after phagocytosis, they live, and neutrophils, after phagocytosis, they die. Two more mechanisms: complement activation, which we will discuss later, and interferon production. Now, I'm not going to talk specifically in very much details about phagocytosis. It's pretty straightforward. You can find it in the main lecture video. So these cells basically engulf the microorganism and destroy it, and that's it. Okay? I'm going to focus on the complement activation and interferon production. So complement activation, and we're going to start with this. So complement is the system of the proteins found in the plasma. They're not in the cells, they're not in the endothelium, they are in the plasma. There are three pathways of the complement, which, for these pathways, the only thing that you need to know is what stimulates them. So we have classical pathway, and the stimulus for the classical pathway of complement activation are antibodies, immunoglobulins, bound to the surface of the pathogen. Then you have lectin pathway. For lectin pathway, the main stimulus are the mannose sugar on the surface of microorganisms that binds to the, so that's bug, binds to the molecules of lectin on the tissue. And the third pathway will be alternative. Alternative pathway is stimulated by a variety of molecules, various macromolecules on the bug. I'm going to call them bug for the simplicity. So these pathways all lead to the same outcome. They converge on so-called C3b convertase. So the C3b molecule then initiates several responses. One response is increased inflammation, and activation of macrophages. Another response is opsonization of the pathogen. That term, opsonization, refers to the process of coating the pathogen. So that's pathogen. Coating the pathogen with the molecules of the complement. Okay, you see how pathogen is covered, and that makes pathogen more attractive, should I say, attractive to the macrophage. So that would be macrophage consuming it. Okay. So it's like adding ketchup to your meal to make it more appetizing. And the last outcome is so-called membrane attack complex. It's the formation of the pore in the covering, in the envelope of a pathogen in the plasma of a cell, of a bacterial cell. That pore, basically a hole in the cell, will lead to the cell lysis. So if you know this, you should be absolutely fine. Last bit about innate immunity is interferons. So let's draw a couple of cells. So this cell on the left, here's the nucleus, which is important, and here's the nucleus as well. So this cell on the left gets infected. That infection stimulates gene expression from the DNA in the nucleus. And that gene expression essentially leads to the production of, mRNA and then protein. And that protein is interferon. Interferon alpha or interferon beta. It depends, type 1 or type 2, it depends on the pathogen, it depends on the cell. The point is, infected cell starts to produce interferon. Interferon is a protein. So therefore, it's a large water-soluble molecule. It cannot cross the membrane, so it has to bind to its surface receptor. Okay? So that interferon here binds to the surface receptor on uninfected cell. It doesn't really matter, on some other cell. Binding to that receptor initiates intracellular signaling, which stimulates the expression of interferon-stimulated genes, ISGs. So basically, those interferon-stimulated genes, they induce some kind of a protective response. It can be apoptosis, it can be mounting up certain antiviral or antibacterial mechanisms like destruction of the RNA or DNA. The main point that I want to make about this story is that you have an infected cell that starts to produce that molecule interferon. Interferon serves as the kind of a warning sign for other cells that may not be infected yet. Okay? So these cells can mount an immune response.